go ahead and introduce our next researcher. This is Dr. Gopal Thinakaran. We are extremely pleased to have him. He is not far from where I'm located at the University of South Florida. And he has got a very interesting project. I'm going to try to read the name, and then he's going to explain to you what all these words mean in the next few minutes. But basically, he's dealing with how do we deal with the genetic system. What, in addition to that, award is going to be for $250,000. It's named for John Trojanowski. John was a member of our review committee for many years and passed away a couple of years ago. And little did we know when we named this grant the John Trojanowski Award was that he knows him personally, and he's going to share with you a little bit about his knowledge of Dr. Trojanowski. And so, Dr. Thinakaran, we're excited to have you today.
It's a pleasure for me to be here to present some of the work and also meet with you, the amazing work that you all do. Take your time to come over here and meet and raise funds to support our research. So I'm fortunate to work in this building in Tampa, University of South Florida. This is a Bird Alzheimer's Center Research Institute. I'll tell you a little bit about that. So first, it's extremely meaningful for me to have this award named after Professor Trojanowski. So I was fortunate enough to meet John a few times. I understand he did a lot of work for CARD Foundation as a grant reviewer. So that's a picture of John, 2004. There's one John in the lab, and there's one with John and his wife, Virginia Lee. So the quote I have here was published in the journal Nature shortly after John passed away. This is a compilation of different quotes, thoughts, memories from colleagues around the world. And it truly captures who John was. He was a legend, a leader, he was a giant in the fielder, he was an amazing human being. And so I'm extremely thankful to have his name appear in my CV from now on. And thank you, CARD, for that. I met John a few times. The thing that I remember about him was how he would take the time to help others, whether he knew them or not. So a couple of examples. When I was in the process of getting promoted and getting tenured at the University of Chicago, we were in the middle of a change of deans. When deans change in institutions, they like to shake things up. I was right in the middle of going through my promotion. So the new dean coming in wasn't sure whether he should help me or not. And I was in the process of looking at positions elsewhere. And the new dean was, or incoming dean was a pathologist, GI pathologist. He goes to a pathology conference, he listens to John. Now John is a neuropathologist, I was a cell biologist at the time. And so we've seen each other, said hi to each other, but John didn't know what all I had accomplished or what my ambitions were, what I was looking for. But the incoming dean asked John, I know you're working on Alzheimer's disease. Do you know about this guy, Gopal? John was so sharp, he figured out what is going on. And so he simply told the dean, Gopal is a good guy. If you don't take care of him, we'll try to recruit him. So then, the incoming dean starts at the University of Chicago, calls me in for a meeting. I thought I was just going to get the papers to look at. Instead, the dean pulled out the notepad and said, tell me what you want, I'm going to give it to you. I had no idea. So then he says, John Trojanowski says hi. And I thought, well, that's kind of sad, strange. I know John, but not that well. So anyway, a few days later, John called me and said, so did you get all you needed? Because I put in a good word for you. I had absolutely no idea. So that was one. The other thing I remember was just observing him and learning. In a particularly contentious scientific talk, there were back-to-back talk by two people that really were controversial. One didn't respect the other or the finding. Basically, one of them scooped the other in a story. And there were some harsh words. At the end, we're walking out. John walked up to the person who was not nice, and he said, "Look, what you did was wrong. Okay, you won the competition, you scooped the other person. And going up before the other person giving a talk and saying, bravo, look what I did, that's like stabbing the knife. But you started to twist the knife. Poor guy, you gotta give him a break." And I was just listening to him standing in line behind him. And then I realized what a decent man he is. Because what he was trying to tell, this is actually somebody he was collaborating with. So he was telling the advice to the person saying, what you did was wrong, you shouldn't have done that. Normally people are just like, hey, you did a great job, you stood your ground. And no, he was basically saying, look, you beat the other guy, but you shouldn't be doing this. So I learned that as a life lesson from John. So with that, continue. So this is the institute I work. I moved from the University of Chicago 4 years ago to Tampa to head this institute. We have a beautiful space for research and clinics and clinical research. This was built by funds from the University, from Florida State, but now it's administered by the university. We have 3 missions. We have a research, discovery research. I'll show you in a second. We have a Bird Alzheimer's Center with Dr. Smith as the director. We see about 1,200 patients at the clinic, and we are part of every major phase 2, phase 3 clinical trials in the field. We also have, what is unique, is a caregiver education program with the director Eileen Poyle. And I know many of you are touched by Alzheimer's disease. I strongly urge you to take a look at this podcast series that Eileen has recorded. We have over 100 of them. We have, I think, 1,700 or so individual listeners from just about every country around the world touched by this podcast. So if anybody wants to have more information, I'm happy to talk to you about that. In terms of the research, we have 3 floors of research labs and neuroscience-related shared core facilities. We're doing really well with the funding from federal grants, but philanthropic efforts and grants like the CART Foundation really helps us explore new areas that are not prime time or they would not be appreciated by NIH for funding because they want to fund things that are absolutely going to work. Whereas private foundation want to see how we can expand into new areas. So I'm very grateful for that. With that, a very brief introduction to Alzheimer's disease. As the previous speaker mentioned, the field was working with the idea that the 2 hallmark pathology of Alzheimer's describes senile plaques and tangles are the driving force of the disease. Incidentally, John Trojanowski was the one who said that tangles are made of tau, and that was his big discovery. That was years ago, before anybody really realized that tau had anything to do with Alzheimer's disease. And so we are, as many of us who started to work on the disease in the early 90s, much of my work focused on what constitutes senile plaques and tangles. And so in general, the idea is that toxic proteins like amyloid and tau, now there's a larger number of proteins. These accumulate, and as we age, they accumulate, and some individuals it accumulates more, and the reason is some of us carry disease-causing genes, risk factor genes, or have lifestyle choices and exposures that promote accumulation of these proteins. These proteins just don't accumulate themselves. There are clearance mechanisms in terms of brain efflux, innate immunity, metabolism, trying to cope up with these proteins, and then also cellular reaction. Ultimately, what happens is these toxic proteins interfere with the neuronal communication network, leading to loss of neurons, and that's the strongest correlative for cognitive decline and death of the neurons in the long run. So what we do in the lab is to start with what we think are important factors, disease-causing genes, risk factors. And we generate a hypothesis, like what you just heard. CD49 is important for cell-cell communication, so that's a hypothesis, and you want to go and test it. So same way we will start to generate hypotheses. We work with the culture neurons in Petri dish, culture microglia, these are the immune cells in the brain. Once we start to understand a little bit what these proteins do, we'll start to infuse them into the mouse brain. Again, there are mouse models that would make amyloid or tau, and they have cognitive decline pathology and cellular reactions. And so we wanted to see if we could validate some of the insights in mouse models. Ultimately, the goal is to advance therapeutic strategies based on what we study. You got a flavor of the complexity of brain cells. And this again shows you that even though originally people thought amyloid and tau are the toxic proteins, yes, they are, except they trigger reaction in a very complex manner with non-neuronal cells, brain vasculature through blood-brain barrier description, and promoting hyperexcitability. This is where brain activity goes up. So there's a lot of these things happening, and so we need to accommodate or understand and incorporate all these cellular changes to be able to find a cure or advance new ideas towards therapy. With the involvement of all these different cells, now we realize that the Alzheimer's disease can be caused by many, many genes, which is not surprising. Originally, everybody was working with a handful of genes that caused early onset Alzheimer's disease, and it would definitely cause Alzheimer's disease. Just like cancer, certain genes, if you have a mutation, you will have cancer. These genes, if you have a mutation in any of these genes, you would be passed on from generation to generation, and it would definitely cause. Whereas most of us have the second category, which is really ApoE is one of them, comes in 3 flavors. If you have ApoE allele, one is at a higher risk for the disease. In addition, there are rare variants in genes that cause medium risk, and then there are a lot more genes that have a lower risk. So the lower risk genes became apparent in the last few years. What is interesting about them is that they all function in innate immunity, or the brain, preventing brain inflammation. Many of them function. So we did a lot of work in my lab on this gene called BIN1, which is the second most significant gene, risk factor gene for late onset Alzheimer's disease. This is just a summary of what kind of data we would get. Just like the previous speaker, we introduced a mutation in this gene in a mouse model for tau pathology, and then we are comparing mice with the gene of interest, without the gene of interest. You can see losing one gene really blocked all of this pathology in the hippocampus. Neuroinflammation is down, and so the brain here shrinks, has a big hole because neurons are dying. We completely rescued that. So we found a way to demonstrate, okay, this risk factor gene is probably not a good thing. We should be able to block it, and it may have benefit. So from the hypothesis, we move on to the stage. So right now we are working with chemists and some companies trying to see if we can find a way to block it. Now, the grant that we wrote, I just want to introduce quickly. The central dogma in the field is that our genes, which is the DNA, will give rise to RNA, which is ribonucleic acid, and then we'll make proteins. So this is what we have. We pass on to the next generation. Every cell will convert the DNA into ribonucleic acid, which then instructs how proteins are put together. And so this is often stated as DNA makes RNA, RNA makes protein, originally proposed by Francis Crick, who went on to win the Nobel Prize. Decades later, what we realized was that the genome defines the complete set of genetic information, but that's not the only thing. Many factors would influence how our genes act. This is particularly interesting, the psychological state that we are in. High stress, low stress, how much social interaction we have, therapeutic drugs, medicines, microbiome, these all could give us a benefit. Even though we have a bad gene, if we did all these, we would have benefit. On the other hand, disease exposure, toxic chemical, drugs of abuse, these could change in a way that things can go bad, right? How does this happen? How do all of these factors influence the central dogma, DNA, RNA, protein? Turns out, the way it works is there's another layer of refinement called epigenetics. So this really means on top of genetics. So what we do, what we eat, how we live our life can imprint in our genes good or bad things, which we then pass on to the next generation, to our kids, our grandkids. And so there's a lot of interesting work goes on in studying the stable changes. And so the changes in DNA predominantly are DNA gets a chemical called methylation, or histones or proteins that keep the DNA clustered or open. They get modified, and also RNA gets modified. So the RNA modification is called epitranscriptomics. So now DNA makes RNA, but what we experience in our life would add chemicals to this RNA. That would then be there, templated, and determine how our cells, our body, our brain can react to disease conditions, to health, and so many aspects of our life. And so basically in the RNA, what you get are these small modifications. Adenosine gets modified to N6-methyladenosine. It's just a one chemical, but it's a whole host of proteins that regulate this addition. There are writers that add the chemical, there are erasers that remove, depending on our lifetime experience, and there are readers that would detect the presence of this chemical and then choose whether the RNA should be made into protein or not. And so there's a very complex mechanism going on here. There are a lot of players involved in this, and when we looked at the published data, this was recently published from human patients with Alzheimer's disease or controls. What they found was there were about 1,415 RNA that had very high modification with this methyl 6A. Out of them, 214 were associated with Alzheimer's disease. So that was the number one association by disease of this RNA. So that told us, however this happens, these modified RNA play a role in Alzheimer's disease process. So let's take a look at this. That's how we started. We looked at all the players. I mentioned writers, erasers, and readers. We looked at the expression of all of these proteins, again, published literature, databases, comparing Alzheimer's individuals with the healthy controls. We noticed there's a group of them that were lower in healthy controls and higher. Just like the previous talk, CD49 was lower in the control, Alzheimer's patients had more. Same way here, there's more. And this is a quantification in a large number of individuals. So we decided we got to look at this. And so we proposed to work on one of the readers, YTHDF1. So the goal is to ask, if you remove the DF1, is it good or bad? So we did a large cohort of animals and did some preliminary data. What we found was removing DF1 didn't really change too much for amyloid pathology, but it did a lot for tau pathology and how the brain reacted to tau pathology. And so this is what we propose to do. We're going to generate mice that normally would go on to make human Alzheimer's disease, a tau pathology. We're going to remove the DF1 and ask how it affects the cognitive behavior. We harvest the brain and ask how it changed the pathology, how it changed the accumulation of the tau and the reaction. So we use a large number of assays for it. The goal is really look at the molecular pathology and cell biology and gene expression to get a comprehensive picture. Does the loss of DF1 reduce Alzheimer's tau pathology? If so, how does it do it? And the second part is a little bit more complicated. As I mentioned, over 1,500 RNA got this modification, but 215 were associated with Alzheimer's disease by just looking at yes or no, it plays a role. But now we remove the reader. Removing the reader means you have a room full of RNA, some have this chemical. A reader would be a person coming in and say, okay, everybody with this particular chemical, follow me. We're going to go translate you into proteins. And that's the job of the reader. We remove the reader. And so there's no selective advantage for the M6 modified RNA to be translated. Now, the end result, we see there's less pathology. That tells us that having the MCA is kind of bad, because once we got that out of the equation by removing the reader, the mice were a little bit better. So now we want to know what those RNA were translated by the DF1, because they all promote the disease process. Now we hope that this study will identify the reader, which RNA it's binding. This is a sophisticated analysis made possible by recent advances in large-scale DNA sequencing. It's called next-gen sequencing methods. And so we'll be able to identify which RNA and where the motifs are. In other words, where the modification is. This will then allow us to name additional targets for further exploration, trying to block these M6-mediated promotion of the pathology. With that, to remind you again, hopefully this funding from the CART Foundation will allow us to expand the number of candidate genes and proteins that we could study in the future. With that, I want to show you the lab group that we work with and the funding. Shui Wang is a postdoctoral fellow who's spearheading the research effort, and he'll be the one funded by the CART award.
My understanding that you're doing groundwork to build on so that other people can see where maybe other therapies could work.
That is correct. So, you know, we are riding on the shoulders of giants who came before us, right? John Trojowski said tau is important. And likewise, my colleague in the University of Chicago, who pioneered the method for identifying the modification, chemical modification, he published a paper and he found this 1,500 RNA that are modified. He shared with us the list. And so we asked which disease, you know, are these things correlating to? And then we realized it was correlating to Alzheimer's disease. Another colleague gave us the mouse where they removed the DF1. And so we are basically getting help from the others. So my goal is we will generate a list of RNA that are different when we remove the DF1, and then we'll publish it, and that would then help others to pursue. We would also pursue, but it will help others to pursue. So it's a way of expanding the science collaboration. Thank you.